Today I'm going to attempt to help you understand a little bit better about the details of neuronal signaling. And in order to understand this, we need to know a few things. We need to understand membrane potential. We also need to understand the role of various channels in neuronal signaling. So within the channels, we have two different types of channels. We have the ligand-gated channels, and we also have voltage gating channels. And both of these are important for different reasons in sending a neuronal signal from one neuron to the next. We also need to talk about pumps, which restore the gradient that occurs after these signals have occurred. So our story here is going to start in the dendrites. And so the dendrites are the part of the neuron that is receiving signals from adjacent neurons or sensory touch and so forth. And so at this site, we have ligand-gated channels. And so these ligand-gated channels will bind to a ligand, and that's what causes them to open. So in the presence of a ligand binding, they are going to open. In the absence of ligand binding, they will be closed. And so one of the other things you have to understand here is that the resting membrane potential, the membrane potential that I just spoke about before, is negative in a neuron. In fact, it's around minus 70 millivolts. And so what that means is we have more negative charge total inside and more positive charge outside. You also need to understand certain gradients. So sodium is high outside. There's more sodium outside the cell than inside. So it goes from high to low, from outside to inside. On the other hand, potassium is high on the inside, and therefore it's higher inside than outside. And so if this channel here, this ligand-gated channel that binds to a neurotransmitter, which might be secreted by an adjacent neuron, if that opens a sodium-gated channel, that's going to cause sodium to flood inside because sodium is moving from high to low. All right. On the other hand, if this is a potassium channel, and therefore it binds to a ligand that opens the potassium-gated channel, potassium is going to flow out, because it also moves from high to low, but it is higher concentration inside than outside. So if we zoom back out a little bit here, we know that this really involves changing the membrane potentials. So the membrane potential, that charge across the membrane, as I said before, it's starting right around minus 70. And so when we're at rest, that neuron is at rest, it's not changing, it's staying right around minus 70. But if we've opened any of these ligand-gated channels, for example, if we open a potassium-gated channel, when potassium is flowing out, that is going to reduce our charge inside because we're losing positive ions from inside to outside, so that reduces our membrane potential. On the other hand, if it's a sodium-gated channel, ligand-gated channel that's been opened, sodium is going to flood in, and with that positive charge flooding in, we're going to raise the membrane potential a little bit because we've just gained positive charges inside the cell. Now, if we focus in a little bit further down this cell to what's often referred to as the Axon Hillock, right? Here we also have some channels. So we also have sodium channels, and we also have potassium channels. But these are not ligand-gated, these are voltage-gated. And so these are going to have a voltage at which they open. And we usually refer to this as the threshold. So the threshold is the voltage at which these either open or close. And most of these are going to open or close right around minus 40 or minus 50. All right, and so what that means is any membrane potential below this, those channels are going to be closed, and above that threshold, those channels are open. So if our ligand-gated channels have opened enough sodium channels or enough other channels that cause this membrane potential to reach that threshold, these voltage-gated channels are now going to open because we just moved from below that where they're closed to above it where they're open. Now here's the important thing. So when sodium voltage-gated channel opens, sodium again is going to flood in, and that is going to raise our membrane potential. When potassium channels open at the same threshold, potassium is going to flood out, and that's going to reduce our membrane potential. But the important fact here is that the sodium-gated channels are faster at opening than the potassium. They open at the same time, but the sodium-gated channels open faster. And so that means we have this brief period of time when sodium is flooding in really rapidly, and therefore our membrane potential is going way up quickly. Sodium channels, however, also have this inactivation gate. And what this does is as soon as this opens, it starts to close. And so it can take that open channel where sodium has been flowing through it for a little bit of time, and that gate will then swing across and close it and inactivate it again. There has been time for some sodium to flow through, but then it stops. So what's happened is our sodium is flooded in, and we have depolarized up to here, but then the sodium inactivation gate is closed. And right about now, and remember, we're talking milliseconds, so this is extremely rapid, but now the potassium channel has caught up, and potassium is starting to flow out. So now we have this time when sodium is no longer flowing in, potassium is flowing out, and so we are repolarizing. We're taking those positive charges back out in the form of potassium. And so when we hit this threshold again, both of these are going to close because now we're below that voltage gating where they open. And so they both close, but the potassium channel closes a little bit slower as well. And so a little more potassium is flowing out until we basically hyperpolarize, and eventually we'll reach back to this resting membrane potential. So if we look back here at the axon as a whole, what we find is that this is happening all the way down this axon. So we have a whole bunch more sodium channels kind of lined up along here, and these are voltage gated, and we have a whole bunch more voltage gated potassium channels lined up. And so in this brief period of time, while this was happening here at this very first area, this caused a loss or a depolarization. And so that depolarization basically is going to open the next voltage gated channels. And so as soon as those next voltage gated channels open, our sodium floods in and our potassium floods out. And that causes depolarization for the next one, floods in, sodium floods in, potassium floods out. And this whole thing is happening all along the axon, really quickly down, and we call this the action potential. So this electrical signal that started at our dendrites has, if it's hit threshold, has started the action potential, which is going to move irreversibly all the way down the axon to the end. And so now, if we take a little closer look at the end, we need to add one more player. So we now have calcium channels. And these are also voltage gated, meaning that they open at the same threshold. So when that action potential reaches here, they are going to open because they are depolarized. And so calcium is also high out of the cell and low inside the cell. So when those calcium channels open, calcium floods in. And their main role is to activate these neurotransmitter vesicles. So all these vesicles just waiting to be released at the end of these axons, they are signaled by calcium flowing in. And calcium is going to cause them to release those neurotransmitters out to the synaptic space between our two neurons. And if there are dendrites of the nearby axon really close, those dendrites are going to have ligand-gated channels. And these neurotransmitters may bind and activate those ligand-gated channels. And if in doing so, this next neuron reaches its threshold, it will send an action potential as well. So basically this whole thing happens again as these neurotransmitters signal are released from one neuron to the next and signal on down. Now, if we zoom back out, the last role that we need to talk about is the pumps. So if we fire this neuron over and over again, eventually we're going to lose our sodium and potassium gradient. Our sodium is going to flood in, our potassium is going to flood out, and eventually there will be even amounts on both sides of the neuron. And at that point, we are no longer able to send an action potential. This actually happens if you fire a neuron way too many times. It's called numbing. It becomes numb if it fires way too many times without being able to reset. So how does it reset? Well, we have, throughout the whole area here, we have the sodium potassium exchanger. This uses energy in the form of ATP to basically pump the sodium back out and pump the potassium back in. And it does 3 sodiums and 2 potassiums per time that this pump works. And this is restoring the gradient so that this neuron can continue to fire. In doing so, we have restored the resting membrane potential. We have also restored the gradients that allow the neuron to fire when the voltage-gated channels open, so that sodium floods in, potassium floods out, and this whole cycle of depolarization, repolarization occurs so the action potential can happen, and one neuron can signal to the next.